Now, going in even more microscopic in skeletal muscle fibers, a little bit more information about them. They're very long. They can be very long. Actually, they can be up to up to 12 inches. That's a pretty long individual muscle cell. And the way that happens is you have embryonic cells, so mesodermal cells. This is part of your embryonic compartmentalization, I might say, which we'll call myoblasts because you're familiar with the word myo and blast means to build. The myoblasts actually get together. So you see this if you look at the images, and again, the image from your book, the one that's labeled A, it's in the upper left. You have individual myoblast cells. These are embryonic cells. They will fuse together to form what we consider a immature muscle fiber. So eventually this is going to become a muscle fiber because it's going to become a little bit more organized, and we're going to introduce other parts to it. But individual cells get together to form a muscle fiber, and in this formation, you have the nuclei of each of the individual each of the individual myoblasts remaining. So one of the features of the second feature of skeletal muscle fibers is the fact that they're multinucleate. They have many nuclei in one individual fiber. And this is very important because muscles are constantly working. That's their job. And that means they are always in need of coding for proteins, and they are always in need for carrying out metabolic processes. And so we know they always need oxygen, nutrients, and then to get rid of waste products, as well as coding for proteins, which we know that DNA is found in the nucleus, and that is the framework for the proteins that are made. So what happens with this immature cell, if it's going to become a skeletal muscle cell, it's going to become more organized into these striations. And the striations are striped appearance under the microscope, and with the image up in the upper right, which is blown up more than our microscopes can work, but not really that much, not as much as a lot of images you've seen in your textbook. You can see these striped appearance, and the striped appearance is because of the repeating units, which we call sarcomeres. And the repeating units called sarcomeres have specific design to them that create this striped appearance under the microscope. So kind of the characteristics of a muscle fiber, or particularly a skeletal muscle fiber, as opposed to cardiac or smooth muscle, and we'll talk about those at the end of the lecture. And actually, I'll just kind of put a little compare and contrast in here. Smooth muscle have individual muscle cells together with individual nuclei, and cardiac have the striation, so they have the design is very different with regard to cardiac muscle than skeletal muscle. Skeletal muscle mature, the mature muscle fiber is going to have a membrane around it, just like every cell that you learned about in previous chapters has a cell membrane. This cell membrane is unique to muscles, so we call it a sarcolemma. And what happens with that sarcolemma is you have a transmembrane potential, which every single cell membrane has a transmembrane potential, and that means the ability to actually move an electrical impulse from the outside of the cell to the inside, and basically the outside of the cell membrane to the inside. And what you have is a positive charge on the outside of the sarcolemma and a negative charge inside the cell membrane. And with that, that dynamic sets up the ability for there to be an electrical impulse that can come along and cause this muscle to contract as a unit. So keep that transmembrane potential in your head. And if you remember back the picture before, you have these units repeating. So you have these muscle fibers repeating in the individual muscle, and we're still thinking about the bicep. The bicep muscle on your upper arm. And what happens is this sarcolemma surrounds the liquidish or the liquid where you have organelles, so the cytoplasm of the muscle fiber. So you have this liquid environment, and that allows for those ions to move from the outside of the muscle fiber to the inside of the muscle fiber, and that begins the muscle contraction. So that begins the muscle contraction. Now, with a muscle like the bicep muscle on the upper arm, biceps brachii, I was trying to use the entire word, that contraction needs to happen simultaneously. So though all of those units that make up the bicep need to shorten, need to contract all at once. And this happens because that membrane potential is going to be passed through the entire muscle via a structure called the T-tubules, the transverse tubules. And you see those transverse tubules on this image as kind of an ochre color. You're going down to the bottom image, and what we've done is we're getting smaller and smaller. At the top image, you can see they've pulled out one of these units. Those are all fascicles, and we pulled out one of the units, and of those one units, you have the T-tubules around around one section. Those T-tubules will take the electrical impulse from the sarcoplasma, distribute it through the entire muscle, and the way this gets distributed through the entire muscle is the T-tubules work with the sarcoplasmic reticulum, so the bluish portion on your image. And that bluish portion kind of wraps around, as well as the T-tubules wrap around each unit of this muscle that is going to contract all at once. So it allows that muscle to contract simultaneously. So it's the transmembrane potential moving on into the sarcoplasma or the sarcoplasma area, and then from there to the T-tubules distributing it throughout the entire sarcoplasmic reticulum via these ends of the sarcoplasmic reticulum, which they call terminal cisterna. So the T-tubules, although the two structures don't intermix, the electrical impulse is able to pass over. And then with regard to a little bit more information, myofibrils, so we're down to the smallest portion that we can look at, are subdivisions of the muscle fiber. They're made up of bundles of proteins called myofilaments, and there are basically two categories of these myofilaments that act on each other and are responsible for contractions. You have thin filaments, which are made up of the protein actin, and thick filaments that are made of the protein myosin. And so actin, thin filament, myosin thick filament. And this picture doesn't come from your book, but that's just because your book didn't put it all together in one shot, and I wanted it that way. But what you have is the muscle, then going into the fascicle, then into what you can see, the sarcoplasmic reticulum, you can see the T-tubules, you can see the termina cisterna, and then we've pulled out even further from that, and we pulled out a myofibril, and then from the myofibril we pulled out one myofilament, which is made up of thin and thick filaments. So a myofilament made up of thin and thick filaments, all made up of proteins that have the ability to contract. That's the organization. And with this is just kind of a rest for your brain and also a blow up. You can see the T-tubules, the ochre color, and you can see the sarcoplasmic reticulum. You can also see the expanded end of the sarcoplasmic reticulum on this, which is the terminal cisterna. The two ends of the sarcoplasmic reticulum that are sandwiching the T-tubules, this is what's considered the triad. And this triad, as your book talks about, is what's responsible for distributing that electrical impulse throughout the entire muscle, at the larger muscle unit that we consider and we learn and we call a particular name, like the biceps brachii. The sarcomere, which I already talked about, I'll get started on that and probably pick it up on the next one, is the contractile unit. It's the sarcomere that is where contraction is housed, I guess you might say. But what you have is millions of sarcomere, one right after the other. And it's the sarcomere that gives, and the organization of the sarcomere that gives that striated or striped appearance, because you're repeating those under the microscope. So they are considered, sarcomeres are the contractile units of the muscle. This is where the actual movement is physically happening, and you just amplify that movement in order to create what we need to actually get from point A to point B. You have alternating dark thick filaments, which we call the A band, and thin filaments, which we call the I band. So you can see that, here you have thick filaments, the A band, and they're in this artist drawing, they're the purpley color. The I band, which are the thin filaments, are the wavy line, and they alternate, and that creates the striations. You have a line which we considered the M line. It's at the center of that thick filament band, that A band, and that has thick filaments but no thin filaments. So you have a certain portion of the A band where there is only thick filaments, and that's an M line, which is the center, and the area is the H band. The zone of overlap is where thin and thick filaments can overlap and do overlap when this muscle is contracting, and we consider that the Z lines. So Z line is where you have this zone of overlap where thick and thin filaments overlap. The I band has Z lines at the center. So there is a place where they can overlap, and that is up to the Z lines. We'll pick this up at the next one.